Welcome to another lecture. This time we're talking about phytochromes. So this is going to be a pretty long lecture, and it's also a lecture about a topic that is somewhat complicated. It's not really that straightforward. There's a lot of moving parts. There's a lot of stuff that's still not fully understood. And there's some things that seem contradictory at the beginning. I'll try to point things out as we go, where things seem to become difficult to understand. But there's a lot known about phytochrome, and there's a lot of stuff that's very straightforward about phytochrome as well. So I think you'll appreciate the things that are known. We'll look at the data to see how we know what we know, and then we'll take it from there. So I decided I'll start sort of with a summary of where this is going. And then maybe you can use that, at the very end again to look back and see, what the lecture was about. So what I will tell you about in this next hour or so is that phytochrome is a light receptor. It's a plant light receptor, specific in plants, also in algae and some bacteria, not in animals. Generally speaking, phytochromes can be activated by red light, but also by far red light. Far red light meaning red light that's in the longer wave spectrum. So an ultra far red light that's not really visible in the visible spectrum for humans. And it can be reverted to the inactive form in the dark. And some phytochrome types are also inactivated by far red light. So here you see the first sort of contradiction where I said it can be activated by red and far red light, can also be inactivated by far red light. So I'll point out why that is and under what conditions that is. So phytochrome acts as a kinase, so it can phosphorylate things. That's what kinases do, and that seems to be light independent. So it doesn't have to be activated by light in order to act as a kinase, but it does have to be activated by light in order to do certain other things under which conditions it acts as a kinase. I'll point that out as well as we go. So just remember, for now, that is a kinase. And phytochrome can help degrade transcription factors. So some of them are called phytochrome interacting factors, PIFs, via phosphorylation and then subsequent ubiquitination. You may remember from cell biology that ubiquitination is one way how cells get rid of proteins by tagging on this ubiquitin tag. And then once there's a ubiquitin tag on it, which is a small protein, then that protein is sort of targeted for that degradation in the proteasome. So it's sort of the kiss of death, people sometimes refer to. So that's how phytochrome works. Some phytochrome types are degraded by light exposure. That's another one of those, right? So it's light activated, but at the same time, it can also be degraded by light exposure. How is that going to work? Well, what that really means, and I'll point that out as well later on again, is that when phytochromes are activated by light, they do what they do, but then after a little while of being in the light, they sort of fade away. And so that's a good idea because that means it can do something, but it doesn't do it forever, right? So it's a light response, and then if there's no more light, then to reactivate other phytochromes, then that light exposure can also get rid of some types of phytochrome. But you also hear me here saying some types of phytochrome, because there's some that are labile and some that are more stable. Phytochromes are encoded by a multi-gene family. So that means that there are many genes that encode phytochrome genes that are producing phytochromes. And exactly what phytochromes look like, you'll see in a moment. And then phytochromes act in combination with other phytochromes. So it's a multi-gene family. There are multiple ways to make a phytochrome, and multiple types of these phytochromes can interact with each other and in combination. And so if you have various combinations of phytochromes, they can have various types of responses. And each type and the combination of types of phytochromes can elicit these different physiological responses. And some of them are categorized in certain ways that I will also point out later as either very low fluence, or high irradiance responses. that basically describes how much light, red light or far red light, is needed for a certain response to occur. So dim light, sort of not so dim light, and then really high light at for a very long time. All right, we'll come back to that, to all of that. Maybe look at this slide at the very end, sort of to remind you of what the lecture was about. Okay, I'm going to start with this really old experiment. I think it's from the 1930s, where somebody started doing these experiments and they observed things. And that's really how phytochromes were best understood, by observing certain things and then looking to see whether that was a red light, far red light response. And then if it was assumed that it might have something to do with this red light receptor called phytochrome. So what happened in this response, in this experiment, was that lettuce seeds were sown in the dark, and then they were given some red light treatment. So red light was turned on, and after a little while, the researcher came back and saw that the seeds had germinated. So we have the light condition and the germination rate. 2 pluses means vigorous germination. Well, what happened when a second experiment was done, in which these dark-grown lettuce seeds, or dark-sown lettuce seeds, I should say, because they hadn't germinated yet, were first irradiated with red light and then irradiated with far red light, so of a higher wavelength. You may call it infrared light as well. When that occurred, all that germination did not happen. So they did not germinate. There was no germination. So you go in, you turn on the red light, and then right after that, you turn on the far red light, you go out of your dark room, and then you come back a week later and you see how many have germinated. Well, it turns out none if you do red plus far red. Well, if you do red plus far red plus red afterwards, within 5-10 minutes or so, and then you come back after a week of letting them germinate in the dark, they notice that indeed germination occurred. And so you can do that again with a far red light, and there's no germination that occurs. So it seemed that there was some sort of a switch that you could toggle back and forth. You can either turn the response on, or you can turn the response off, or either on or off and on and so forth. And it appeared that that was light quality dependent. So red, far red, it's the quality, not the quantity, the quality of the light. And light quality can initiate a developmental response. This reversal, so that the reversal of development, can only happen, though, within a certain amount of time. I said here, so this happened within 5 minutes on, 5 minutes off, 5 minutes on, 5 minutes off, and then a week of delay while these seeds were germinating. If you turned on the red light and then you came back the next day and you turned on the far red light and expected to see no germination, that was not happening. So if you waited too long between the two treatments, so in other words, between turning the light switch on and the light switch off, or this developmental switch, I should say, then at some point there was a commitment to the response. So the plant had committed to the red light response, and far red at that point was no longer able to turn the response off again. So this can be reversed, but only before commitment to the response occurs. All right. So let's take a look here at some phenotypes. So here we have a plant. This is a bean plant, and it was grown in the light. That is called, in botanical terms, a de-etiolated state. An etiolated state is the state of growth in darkness. So if you go to the garden and you turn over a big rock and you see that underneath there are some dandelions and they're sort of yellowish, and then there's no green leaves, but there are some leaves on there. Well, that is an etiolated plant, and it's because it didn't get any light. It still grew, but it didn't get any light, and so it developed very differently than a plant that developed in the light. Now, most seedlings germinate underground in the dark, and so the first phase of their growth is in the dark. And so that's completely normal for a plant to have an etiolated growth phase, and then as soon as the seedling pokes its head through the soil and sees some light, that's when it switches from the light growth to the dark, from the dark growth to the light growth. And so it switches from what's called scotomorphogenesis, growing in the dark, developing in the dark, to photomorphogenesis, growing in the light. It makes some sense if you think about the energy resources that the plant has at the point while it's growing in the dark. It can only use what is packed in the seed from the mother plant. So by the time that that's all used up, there is no more energy, right? So the only energy that can come to the plant at that point when the seed resources are used up are from sunlight. So if the plant hasn't reached sunlight by then, it cannot produce any more energy and then it will die. So it makes good sense for the plant to grow fast and furiously, very quickly through the darkness, through the soil, until it reaches the light, and then not to grow fast anymore, to become smaller. I mean, it won't shrink, obviously, but the growth rate will not be as fast anymore. And so the plant then slows down its growth, and instead it starts developing leaves. It produces chlorophyll, and then with the chlorophyll, of course, it can do photosynthesis, and then it can sort of relax and start a slower life where it has access to light and therefore to energy. Okay, let's go back to the phytochrome. So what does a phytochrome look like, chemically speaking? So phytochromes are proteins that are linked to a chromophore. So proteins that are linked to something else are often referred to as apoproteins. An apoprotein is just a protein, just a regular protein, and it has something else on it. And in this case, it had this chromophore on it. So this chromophore that is linked to this particular cysteine of this polypeptide, well, it goes on here and here, and it's a protein, it's a long protein. That together is the phytochrome. So it's an apoprotein plus a chromophore. And this particular chromophore is called phytochromobilin. You see that it's made up of these four rings, carbon rings, and then linked with a thioether linkage to the cysteine here. And now the question is, how does phytochrome, how does the structure allow phytochrome to respond to red light or to far red light? Well, what happens when you shine red light on this structure is that the energy that's in red light converts this cis bond here to a trans bond. So what happens is basically that the D ring flips up, it rotates around this carbon here, carbon 15, and moves to an upward flipped position, if you will. And if you shine far red light on it, then this flip switches back. So this ring, this movement of the ring within the chromophore is what basically constitutes the switch of this particular compound. All right, so I said apoprotein plus chromophore. I said this is a linear structure. It's a tetrapyrrole, has these four rings. And red converts from cis to trans. That's a conformational change. That is what allows the activity to start. So here's something that's complicated about phytochromes. And that is that even though this is what you will see in every textbook, even in an introductory textbook, red turns on, far red turns off, that's really not the whole picture. That's not the whole story. So let's take a look at this part here of the slide. So the PR form, that stands for phytochrome red light absorbing, PR, phytochrome red light absorbing, that will absorb red light very quickly, and it will turn that into the PFR form. So here's the PR form, here's the PFR form, and the PFR form, that stands for far red absorbing. So at this point here, this structure is ready to absorb far red light and flip back to the red light absorbing form. At this point, the structure is ready to absorb red light and flip to the other form. And you see here, this is a bright red arrow. This is sort of a dark red arrow to indicate the different light qualities that will create this versus that response. But you see here that this response is only 85% efficient in this direction and only 97% efficient in that direction. That means that even if you shine red light on this, not all of the PR form will actually become PFR. Some of it will stay in the PR form. And likewise, if you turn the far red light on, not all of the light will, not all of the phytochrome will be turned back to the PR form by far red light. It's only 97% efficient. That's pretty good efficiency, but it means that there's still a little bit of PFR form left even after shining far red light on it. And that explains some things that people have seen, sort of some reactions that don't conform to this nice canonical red light turns things on, far light turns things off idea. So there's a third response that also occurs, and that is the dark conversion in darkness. So it's a very slow response, but during the night, PFR, activated phytochrome, is slowly reverting back to the inactive form. So this is the inactive form, this is the active form. And even that you have to take with a grain of salt. But for the most part, that's true. So inactive, active, but then in the dark, it slowly converts back to the inactive form. So those are the three light conversions that phytochrome can undergo. Red will turn it on, far red will turn it off, darkness will also turn it off in a very slow manner. All right, we'll come back to all of that when we see what these phytochromes actually do. So physiologists love to look at light receptors with respect to the question of where in the spectrum, in the visible spectrum, they absorb light. If you've taken Bio 111 at Puget Sound, you have done an experiment where you extracted chlorophyll, which of course is a light absorbing molecule required for photosynthesis. And you may remember that you took that extract, you put it in a spectrophotometer, and you measured its absorbance at all these different wavelengths that the spectrophotometer could produce. So this is on the x-axis, the wavelength. So assume somebody put an extract of either the PR form in green or the PFR form in blue separately into a cuvette, and then looked at each of these wavelengths to see how much of the light at that particular wavelength, so the color of that particular light, was absorbed by that particular compound. So you see that the red light absorbing form, the PR form, absorbs a ton here in red light. So the overall absorbance in red light at 666 nanometers is the greatest amount for this particular compound. But you see that there's also a little bit of absorbance, significant absorbance here in the blue or ultraviolet area, and that there is actually a little bit over here, I'll point that out again, in the far red area as well. Not very much, but some. And now the PFR form, that does in fact absorb the most in the far red spectrum, infrared spectrum here, but it can also absorb relatively well, not nearly as well as in the far red, but also a little bit in the red, and almost as much as the PR form, also in the blue light. So that already starts to explain a little bit why it is that these reversal responses are not 100%. They're 85%, 97% in one direction or the other direction. We'll come back to that. But if you look at these shoulders here, that's one of the reasons why this is not a complete 100% red response or far red response. So absorption spectra describe the ability of a compound to absorb light at a given wavelength. That's the definition of an absorption spectrum. It's important that you remember that you know what that is, because we will talk about another type of spectrum. It's called the action spectrum in just a little bit. And we'll come back to these absorption and action spectra for a number of compounds in plants not only chlorophyll, but also some other light receptors that you will be introduced to later on. So good idea to just know for right now what's an absorption spectrum. Well, that describes the ability of a compound to absorb light at a given wavelength. And this is how you graph it. All right, and then I had pointed out, and I want to write this down here for you to note, that the red light absorbing phytochrome also absorbs a little bit in the far red, right here, and in the blue, and that the far red absorbing phytochrome also absorbs a little bit, or quite a bit, in the red light and in the blue spectrum, so here and over here. And this explains that phytochrome conversion is not 100% efficient. Okay. All right, so here is, in contrast to the absorption spectrum, an action spectrum. You might have talked about an action spectrum in Bio 111 when you first heard about chlorophyll. So there's a famous experiment. We'll come back to that when we talk about photosynthesis in this class. But essentially, what you do when you talk about action spectra is that you take a plant, not an extract. You take the plant and you're looking at a particular action of wavelengths. So the question is, how good is a wavelength at causing a particular function, a particular physiological function, such as germination? And so what you would need for that is sort of a filter wheel that gives you different colors of the light. And so in this case, people used ultraviolet light, and then purple light, and blue light, and bluish greenish light, and green light of 3 different wavelengths, and maybe even 5 different wavelengths, then a yellow and orange, several red tones, and far red tones. And so these were basically 10, 12 experiments for each of these dots at a different wavelength, so different color light. And each time the experiment was done, they turned on the light and they asked, well, how good can this particular wavelength, is this particular wavelength at creating the phenotype that I'm studying, so germination, for example? Well, it turns out if you use this light only here, this bluish light, you get 18%, 15% to 18% tops of germinating seeds. If you use green light, you get a little bit more. So you get this percentage here of germination. But if you use red light, here is the best level, 100% germination, if you use light at, well, about 660 nanometers. And it turns out that if you use a more higher wavelength red light, then that is no longer the case, that the germination works well. So this here by itself, this line alone, is an action spectrum. It describes the action for germination in this case in this spectrum for a particular response. In this particular graph, you have also an inhibition spectrum. So it also shows you, I mean, you could say that that's an action as well, right? So inhibiting germination. And so they did the same thing, separate experiment, and they looked at the question, how well can you inhibit, maybe after first activating with red light, germination by these particular wavelengths? And they found that only in the far red light, a little bit in the red light though, you can also get inhibition of the previously activated germination of these seeds. So here are 2 responses in this action spectrum. So an action spectrum describes the effectiveness of a particular wavelength on a specific response. For each response, the action spectrum may be different. So when I say each response, I mean germination here, flowering time, greening up of the leaves, expansion of the leaves, growing of a stem, or shrinking, or not growing as much anymore of a stem or an organ. Each of those could be a particular specific response, and the action spectrum would describe the effectiveness of a particular wavelength on that particular response. So the overlapping shoulders that I pointed out in the previous slide, I want to point out again here. So even in far red light, some stimulus of germination occurs, and even in red light, some inhibition of germination occurs. And that is what I told you earlier. After red light activation, only 85% of the PFR form, the active form, is produced. 15% remains in the red, in the PR form, and after far red, it's 97%, and 3% remain in the active form. All right, so this slide tells you about the different types of categories that phytochrome-mediated responses have been grouped into. This is sort of a historic way of looking at responses. It's very common, so you will hear that this is a VLFR or an LFR or an HIR form response. And it may not be so important, with the one caveat that it does tell you something about whether or not maybe just a tiny little bit of phytochrome is enough to stimulate a certain response, or whether you need this phytochrome to be mostly in the active form. So if we go back to the idea that shining light on it will not 100% turn one form into the other form, if you have responses that use certain types of phytochrome, but they only need a tiny, tiny little bit of it, then maybe these 3% might be enough to elicit the response, even if you thought you turned it off, with the light stimulus in the other direction. So that is why categorizing phytochrome responses, might still be useful to describe certain phytochrome phenomena. So let me tell you what they are. This is in your book as well. You'll see these responses. You should probably know what they stand for, what they mean. So there's a bit of definition. Here on the y-axis, the relative response. So how well is the response happening? How much is the response happening? And this here down here tells you about the fluence in log units, the fluence of light. So that is the micromoles, numbers of photons, light units, per square meter. So fluence is the photons per square meter. Basically, how bright is it on a square meter? And this part here, by definition, this area here is the VLFR, so the very low fluence responses. When you have this little bit of light here, then you can elicit certain types of responses. And this little bit of light here equates to not much more than the light that you would get from a starry night. So you go outside and there's some stars in the night sky. That might be enough. That would be enough light to elicit some of these very low fluence responses. That's how very low fluence you need. Then there are these LFR responses, and most of these are far-red reversible. Now, if you think back to the very beginning of the lecture, I told you about an experiment that sort of set the tone for all subsequent experiments in phytochrome research, the one with the germination of the lettuce seeds. Well, that was a classic response. There was an LFR response. So far-red light, reversible, you turn it on, you turn it off, you turn it on, you turn it off. The very low fluence response here, you see that this table says it's not far-red reversible. It's still a phytochrome response, but it's not far-red reversible. And that is, again, because if you turn on phytochromes and then you turn off the phytochromes, or at least you thought you turned it off, but you left 3% or so in the active form, that's enough to turn on these very low fluence responses. And so therefore, you cannot turn them off. You cannot turn them off by the far-red treatment, because even far-red treatment will still leave enough active form for this type of response to occur. And then the last type of response is the high irradiance responses. So the fluence rate, these are fluence rate dependent. And in this case, it's not just the amount of fluence here, but it's the fluence rate that's important. That's why they're called high irradiance response. I have some definitions on the next slide for these light physical terms. The fluence rate takes the concentration of the light, so the brightness of the condition, into consideration, but it also takes the time into consideration. So how long was the light on? Maybe here you just have a burst of light not so bright, brighter burst of light. But here, not only do you have a bright light, but it's also on for a certain amount of time. So this reciprocity, I'll get back to that in a second. So here it says reciprocity applies, reciprocity applies, reciprocity does not apply for the high radiance responses. So what does that mean? Let me write that down for you on the next slide. So reciprocity means essentially that a short and bright amount of light treatment is the same, as far as phytochrome is concerned, as a long and dim response. So you could take, a bright flash, or you could take just sort of a very dim piece of bulb with a little bit of light coming out and just leave that on longer. Then both of those types of experimental conditions would be reciprocal to each other. Reciprocity applies here. Both of them can do the response. With high irradiance responses, that's not the case. They you can't give it a bright enough short amount of stimulus in order for this response to occur. It has to be on for a while. So high irradiance has that time component as well. Let's actually start here. No, let's start up here. So here are some definitions that I mentioned. What is fluence? Fluence is the number of moles per square meter of photons. Irradiance is fluence, it's the fluence rate, because it says it's the fluence here, mole per square meter per second. So time goes in. And then if we start here with a low fluence response, these are the units for the what's considered a low fluence response. They are far rate reversible. And there are some examples that I have here. So for example, we talked about the seed germination. This is the canonical phytochrome response. If you ask somebody about phytochrome who only has a minute to tell you about it, or if you look in a textbook that's not a plant physiology textbook, but maybe just a biology book, they might mostly talk about this reversible, canonical, original response that was first found in phytochrome experiments. So deediolation, hypocaudal inhibition, so that's this growth of the seedling stem that grows fast and furiously first and then grows much more slowly after reaching light. That is a far rate reversible response. Chlorophyll production, seed germination in lettuce and also in Arabidopsis. And then for the high irradiance response, some of the examples are pigment production in fruit. So the fruit hangs on the plant for a long time, several weeks, and the light is on and the sun is out, and so it will produce pigments. Anthocyanin production against photo damage. So like the red colors or the red shades or blue shades, those will require a lot of light for quite a while. And then we have the very low fluence responses, so that those are the ones that are not reversible. See, it only requires about 0.02% of all phytochrome to be in the PFR form. And so, yet again, when I say, this switching from the inactive to the active form, and then back from the active form to the inactive form, which is only 97% effective, well, if it's 3% ineffective, that's plenty for these responses to occur that only need 0.02% of active phytochrome. Okay. All right, let's take a look at some sort of ecophysiological responses. This graph here shows you the photon fluence rate relative to the total of what the sun has to give in a sunny day out in the open. And it shows you here the wavelength, and it shows you the wavelength of daylight. So this is basically the sun. If you were to go out and you were to take a photoradiometer, we have one in the department, we'll probably use that in lab as well. And it tells you, rather than a spectrophotometer where you say, I'm putting these wavelengths in here and then tell me what happens at these wavelengths, it's the other way around. You take a light and you say, what are the wavelengths that this particular light emits? So this light here is the sun, and you see that it has quite a bit of activity here in the 500, so green to red spectrum, of course also in the blue. And then it has this far red drop. That's a natural occurrence. I think it's due to the ozone layer. I should have looked that up again, but there is absorption of far red light, and there's this particular dip. And this bottom line here, that's this dashed line, that is what you see with this spectroradiometer if you go into a forest or under a tree and you ask, well, what kind of light comes through the leaves after the light has been filtered through the chlorophyll, through the green color of the chlorophyll. And you see that not much is left here, in the blue and the red. There's a little bit in the green because obviously the color of the chlorophyll is green and it reflects the green, so you still get some of the green. And then there is this huge amount of this huge amount of light that you get from in this particular spectrum, in this particular part, and that is because the red light has been filtered out, right? The red light has been filtered out by the chlorophyll, but the far red light is not filtered out. So the far red light is not filtered out, the red light is filtered out, and then this dip is really, it's just the same because of the dip, the natural dip of sunlight. So let me point out what is interesting and what is important here with respect to some ecophysiological aspects. And that is that the ratio of red and far red seems to be important for the response that phytochrome elicits in plants. So it's not just so much the idea whether you have only far red and only red, because if you think about this, what plants are out there in nature that actually will receive red light. Nobody goes out there with a red light lamp and shines them on plants and then expects that that's the only way how they grow. So red and far red light are, of course, parts of the natural spectrum. And as it turns out, what is important for plant growth is more so the ratio of these two components to each other than the total amount. So let me point this out here. So in sunlight, if you just go out in the bright sun, in a clearing, no other plants, you see that in the red and in the far red, you have about the same amount, right? It's about the same overall fluence rate of red and far red in the total amount of light. But if you go under the tree, where the red has been depleted and the far red has not been depleted, right? This has been depleted because chlorophyll has absorbed it. Now you get a red to far red ratio that is very different from the one in sunlight. So here the red to far red ratio is about 1, about the same as this. Here the ratio is maybe 10% of this, right? It's maybe 10% of that. So the red to far red ratio is 0.1. And so this is a shady condition, and plants can determine whether or not they are growing in the shade or whether they grow in full sun by using this ratio of red to far red light, and of course using the response that they get from the phytochromes that can read this difference in red versus far red. So down here, I have a few interesting pieces of information that have to do with these ratios in particular conditions. So here's the red to far red ratio, and if you just look at a midday condition, so you go outside midday, this is what you will have. So 1 to 1.2, so it's about this here, right? So this is about the midday condition of just bright sunlight. If you go out in twilight, mornings or night, you see that this red to far red ratio has changed to maybe 0.85 to 0.96, depending on what time of the twilight you are looking at. And sort of intuitively, if you think about light outside during the day versus the morning or at night, especially if you maybe are a photographer, you will know that the light sort of looks very different if you take a picture of something in the morning or in the evening versus in the middle of the day. The colors are quite different, and it's because the sun filters through the atmosphere, and if the sun is right above the observer, then there's a relatively thin layer of the atmosphere, while when the sun comes in from the side, then that layer that it filters through of the atmosphere is brighter, is thicker, and so therefore the absorption is slightly different during the day versus at night. So a photographer knows that sort of intuitively, but you can also physically measure that. And then I have here under canopy 0.1, so this is right here. And then under 5 millimeters of soil, of course, it depends a little bit on what type of soil that is. The ratio is different, so 0.88, so that a plant can see the difference between living under a little bit of soil. This is probably a seedling that has just germinated. It's not growing in full sunlight yet, but it's also not in the shade. Plants that are in the water, so water plants, they have a different red to far red ratio to work with. And then here I wrote north-facing slope, so a north-facing versus a south-facing slope. If you look in the mountains, if you're interested in ecophysiology of plants that grow in the mountains, you can see that there's a different type of population of plants that grows in south-facing versus north-facing slopes of mountains. Right? So they get more sun, they get less sun, but they also get different type of light. So the quality of the light on a north-facing versus a south-facing slope is different. Okay, so let's take a little look at what these different types of responses, red, far red, white light, blue light, UV light, and then these responses that are dependent on the ratio elicit for the plant. So this is from a nice review article that I took from 1995 already. Here's your light, and here's the plant, a plant, and that is producing shade. And so if the plant is growing under this other plant, then it is growing in shade. And you see that it has a shade avoiding response. So it basically grows very long. Makes some sense, because if the plant is growing in the shade, or it's growing under a tree that is, let's say in the forest, that is just starting to leaf out, it's producing shade. Well, the only way for this plant to be successful and produce seeds before it's completely shaded is to grow fast, ideally out of the shade if it can, or flower quickly and then produce seeds and then complete its life cycle before it gets shaded completely by larger plants. That's exactly what phytochrome does. So here you see the shade avoidance response. Early flowering is also something that or flowering time is also something that is affected by phytochromes. Then here we have a plant growing in the light, so it is much shorter than a plant that's growing in the shade. And then there's also directional light, and we'll talk about directional light, what that does to plants. This is a phototropic response. We'll talk about phototropism in a later lecture. So here's the short photomorphogenesis, characteristic photomorphogenesis. Here's the shade avoidance. Here's the phototropic response. And here's the elated response in darkness, long, growing quickly to try to reach the light before running out of fuel. So down here are sort of what you expect from these different types of conditions. So in B, wild type plant growing in continuous red light. So it has sort of a medium stem length. While you have a wild type plant growing in continuous white light with a high ratio of red to far red. So these would be the ones that are not in the shade. That's this here, that's D. A plant that grows in continuous far red light, and I'll show you actual photograph of some plants in a little bit, they never really produce regular chlorophyll. So far red light does not elicit chlorophyll production. So they do something that looks different than this. They do slow down in their growth, but they don't really green up. And then a plant that grows in continuous white light with a low red to far red ratio. This is a long plant, so I wrote long here so that you can see the difference between that and that, which is not that obvious. But those are supposedly the different types of conditions, light conditions. And then I have here a light growing in blue light, which is also short, and a plant that is growing in continuous UV light, and that is also short, but it's also pretty sick. So you'll see that, actually we probably won't talk much about this, but a plant growing in UV light is, there's a lot of damage, and it also doesn't have the right wavelength to produce green, perfectly normal leaves. Okay, so here are some pictures of real plants. So we have the low red to far red, causes the shade avoidance response. We talked about that. So bright light, this is an Arabidopsis seedling. Here's an Arabidopsis seedling in shaded conditions. Not only is it a little bit less dark green, but I think that's not the main part here. So it does look a little bit yellowish, but the main part really is that it grows much, it's much longer, try to get out of the shade, and also flowers very quickly. All right, so that's sort of some of the physiological responses. I'll show you some pictures of physiological responses in just a second, but I want to combine it with sort of a concept that is really important for you to understand throughout the rest of plant physiology class. So, if you need to take a little break, maybe take a little break right now, come back to this in a second. We'll go another 10 minutes or so, I think. But the next part is really important. So if you're drifting off, take a little break and then come back to the next slide. All right. All right, so here we are. This is a technique that is used, that has been used very successfully by plant scientists to understand the physiology and to understand the genetic underpinnings of many, many responses. So it's called mutant screen. So looking for mutants, looking for a plant, an individual really, that has a mutation in a certain gene, which is responsible for a loss, almost always a loss of a function. And so therefore, if that particular mutant has a loss of a function, then the lost gene that the mutant doesn't have anymore must be responsible for that function that the mutant doesn't have anymore. Right? So there's a lot of negatives, double negatives. Let's walk through this. So what people do in this kind of experiment called a mutant screen is they take a plant and they take the seeds from the plant. And this is a little seed container with a lot of seeds. And they treat that all the seeds with a mutagen. That can either be a chemical mutagen, something that destroys specifically certain bases or base pairs or causes changes in the DNA structure that way. Or it can be a piece of DNA that gets inserted into the DNA. That's called a tag, and a DNA tag. We probably get back to that as well. Some bacteria actually do that they insert little bits of DNA into host DNA. And that is frequently done in the lab as a way to mutagenize seeds. But the chemical way is also one way. It's a very simple, very easy way to do. They both have advantages and disadvantages. The point is that you want to break about one gene on average in each of these seeds. But of course, it's random. Right? It's a random process. So not the same gene will be broken in each of these seeds that you have in here. So each seed will have a different mutation. So then you take all the plants, all these seeds, you plant them under screen conditions. So what do I mean with screen conditions? Well, if you think back to, for example, our salt discussion. If we were interested to find a plant that has a certain effect, find a plant that has a mutation which has a certain effect on the plant's ability to grow in a moderate amount of sodium chloride in the soil. Then here's our little box, and there's a little bit of sodium chloride in there. Not enough to kill all the plants, but just a little bit more than you would normally have. And then you plant all these seeds, and each of these seeds have a different mutation, right? Each of them have a different mutation. You don't know where they are, but you just know that you added enough chemical mutagen or UV radiation or other ways to mutagenize the DNA to break on average one gene per genome. So you grow up these plants and you self them. So that's one thing that plants do really well is they self. Some do. Not all of them do, but model plants are often used as a model because they can self. So let's say this plant self-is, and then you take the next generation, the F2, where now that particular one gene that was in the first generation only broken on one of the two chromosomes, after selfing, going through segregation, genetic segregation, you now have some plants, right, 1 to 2 to 1, so a quarter, that are homozygous in their mutated gene. So that mutated gene should be, you should, from each of these plants, you should have, you should be able to have some that have now a homozygous mutation. You grow those up, and then you look again in these screen conditions for plants that are responding differently than everybody else. So here, all of these plants looking just fine. They can withstand a little bit of salt. They might have been mutated, but they haven't been mutated in a gene that is important for the plant's response to salt, salt in the soil. So all wild type go okay, but a sodium chloride overly sensitive mutant is found, and that's this one here, right? Likely lost a gene that protects against sodium. So that plant here doesn't seem to have a particular gene that allows these plants to grow and deal with that extra salt. We talked about salt transporters, right? We talked about salt overly sensitive mutants, the SOS mutants. So maybe this is one of those. So now you go, you come back after a few days or maybe weeks even to your screen, and you say, well, let's look at all these plants. And since you don't know where the gene is and you can't see it from the plant, the only thing that you can go by is the phenotype. And you want to find a phenotype that's easy to score. So this is a very easy to score phenotype, right? Even if you had 10,000 plants that are all looking fine, one guy that looks sickly, that's the one that you would want. So now you take that plant. In this case, you would probably stop growing it on salt so that it doesn't die. But now you isolate the mutant gene, you deduce the protein structure, you analyze the protein function, you really try to get molecularly at the question of what that particular gene does. And how you do all of that, we can talk about at another time. But this process from beginning, from mutagenesis, from the screen to the isolation of the gene, that is called a mutant screen. And the screen about that, the important thing is that, depending on how you grow these plants here in moderate sodium conditions, that is what will sort of tease out the function that you're looking for, right? If you're not looking for something that's sodium, or let's say if you didn't put any sodium in the soil, you would never see the difference because this guy wouldn't be stressed if there wasn't any sodium in the soil, right? So you would have to set the screen conditions such that you can actually find out what you're looking for. All right, so why did I tell you all of that? Because that's exactly what people did to find the phytochrome mutants and the phytochrome genes that are responsible for making the proteins that then have the chromophores that do the switching and the absorption and the responses. So let's look here in this particular paper from 1998. They took plants, wild-type plants here on the left, and these plants are growing in the light, and this plant is growing in the dark. And you see this is a normal response, so this is a small plant, this is a larger plant, and the plant is larger because it is just doing scotomorphogenesis while this guy is doing photomorphogenesis. It stopped growing so fast when it reached the light or when it saw light. Now the phytochrome A and the phytochrome B mutant, these are mutants. These are mutants that came out of a mutant screen where they were looking for plants that just look like this, but they found some that are actually a little bit taller. So they were looking for plants that stood out from all of those tiny little plants. They were looking for some that were reaching their heads out over the lawn of other plants to look for light, essentially. And they think they can't find it. And the reason why they can't find it is because they don't have the right photoreceptors. So in white light, two genes that were identified that were later called Phi A and Phi B have this type of response. But it becomes more easily visible when you use red light as the screen rather than white light. So in red light, the red light response, as we talked about earlier, is sort of a shade avoidance response. It's sort of a, it allows the normal response to be larger, but for the phi B mutant, for this particular type of mutant that was isolated, that response becomes very, very much exaggerated. So the seedling is very tall, it grows very high because it cannot find the red light. And so it appears that the phi B gene is specifically encoding a phytochrome photoreceptor that is red light responsive. The phi A mutant, as you see, it looks just like the wild type. So it seems like the phi A mutant must absorb other types of light, but phi A does not seem to be specifically responding to red light. Okay, so then they did the same thing with these two mutants, and they grew these in far red light. So from the same seed batch, some seeds are growing here, some seeds are growing here, some seeds are growing here, from these different types of mutants. So in far red light, I showed you that earlier in that cartoon. Yes, the wild type will slow down, so it can see that there is light, but it doesn't really green up very well. And the phi B mutant, the one that over here in red light had this exaggerated response, that actually looks just like wild type. But the phi A mutant now looks as if it's in the dark. It can't see any light. It just keeps growing, and it does not look like the wild type. So it appeared that the phi A gene was the one that was specifically conducting the far red response. So on this table, I've sort of put this together from what we saw in the previous slide. So here are the mutants, phi A, phi B, and the wild type. The conditions: dark, white light, pure red, pure far red. You see that this is the wild type response in phi A in red light. Here's the wild type response in far red light. So it doesn't look any different than the wild type. But in the phi B mutant, you get this shade avoidance response in white light. You get a really exaggerated shade avoidance in red light. While in the phi A mutant, you get these small cotyledons and sort of a shade avoidance response in the mutant here, while the phi B mutant has a normal response to pure far red. All right, so what to take away from that? The take home on that is that phi A and phi B seem to act additively to inhibit the hypocaudal growth in white light. You saw that the white light in figure A on the left, the one that I showed you first, does not look normal, right? But it doesn't look like the two far red or red pure experiments. So they seem to act additively. Both of them together have something to give for this particular response. Phi B is required for the normal shade avoidance response. So phi B, the gene, not the mutant, for the normal response. And then phi A is required for the hypocaudal inhibition in far red light. Okay, so just briefly, we will take a look at this probably in class. Here's another tool that molecular plant biologists use in order to better understand how genes work, how genes function. So these are called fusion proteins, and they can be, for example, proteins that are fused to what's called a reporter fusion. So this is the GFP, the green fluorescent protein that you might have heard of before. Originally was isolated from a jellyfish, makes it bright green, and they can be used to track the gene expression or protein movement. So let's see how they do that, and then you'll see later on, how that was done with respect to phytochrome. So if you have your gene, you've isolated it from the mutant screen, and you now know the molecular identity of it. You have the promoter, and you have the gene sequence. So the promoter is the gene switch, you may remember. This is called the native gene. This is sort of the wild type phytochrome gene. If you now make a GFP fusion and you introduce this construct, this is the construct, you introduce that into agrobacterium. Agrobacterium is a natural genetic engineer. It's a bacterium that will take DNA, its own DNA, and will put that into the host plant. But if you first engineer the agrobacterium and you give it something else in terms of DNA, then it will also stick that into the host plant if you infect it with that. We'll come back to that later in the class, to the details of this. But this is how plants often are genetically modified, using this natural genetic engineer, this agrobacterium. So you make a fusion, you take the actual native promoter, and then you lob off the phytochrome gene and you stick instead the GFP gene on that. Now what you have is a GFP reporter for gene expression. So now you can see when this gene is expressed in the plant, it will produce, whenever this phytochrome promoter is on, it will produce the mRNA of the GFP gene, and then that will be translated into the protein. So wherever this is expressed, the GFP protein will be found, and you can look at the plant, you can shine a UV light on it, and that will make that green fluorescent protein fluoresce. And there's a second type of fusion, and that's called a protein fusion. So here's just an expression reporter, just the promoter and the gene instead of that. But remember that this is the phytochrome promoter. It's not just any promoter. Here we have the phytochrome promoter again. We have the GFP gene again, but we leave the phytochrome gene in there. So now this reporter will make the phytochrome gene whenever the phytochrome promoter is active, but it will stick on it, sort of at the end of it, it will have a bit of a GFP that will still glow. So now this is a green glowing phytochrome protein that you're making. That's the GFP protein fusion. We'll talk about this as a way how you can use that to study phytochrome function in class. So hold that thought. We'll come back to that. There's one more thing I want to show you before I'll let you go. And that is that at the beginning I told you that phytochromes act as a kinase, a phosphokinase. I also told you that it acts through transcription factors, and that interaction with those transcription factors allows for the plant to, do its physiological responses. So there's a nice cartoon here that I got from this article that illustrates that nicely, and I think maybe that is how we want to end. So the plant grows in the dark, and it has phytochrome, let's say phytochrome A here. It's not active, and while it's not active, the plant is growing fast and furiously. So how does it do that? Well, it has these photomorphogenesis genes that will slow down the plant and that will allow the plant to produce green leaves and chlorophyll and shorter hypocotyls. But it can't express these genes because this particular protein called PIF3, phytochrome interacting factor 3, sits on the promoter. It inhibits the transcription of these genes. So when the light comes on here, the phytochrome becomes active. You see how it switches from this pink to this red form. Now phytochrome is activated, and when it's active, it can move into the nucleus. It can now act as a kinase, say it's a phosphokinase. So that little P here means that it's now phosphorylating PIF3. So that guy down there that's sitting on the photomorphogenesis genes and saying, no transcription today, that is being phosphorylated by phytochrome in the active form right up here. And when it phosphorylates that turns the PIF3 into sort of a sitting duck for the ubiquitination pathway. And what that means is that now other enzymes come by and they say, oh, you are phosphorylated. That means I will stick on a ubiquitin tag, which is a small protein tag. And when they are phosphorylated and then ubiquitinated, that means that's the kiss of death. For proteins, they go into the 26S proteasome and they get chopped up there and inactivated. Now, PIF3 no longer sits on the photomorphogenesis genes and can become active can allow for the transcription of these genes. And so they become active, they allow the plant to green up and do responses in response to light, mediated through phytochrome. That's one type of response that is molecularly well understood, where all the players in the signal transduction pathway are known, and where genes are known that then will lead to this particular response. So sort of in summary here, PIF represses the light response genes in the dark, the light activates the phytochrome, then phytochrome moves into the nucleus, phosphorylates PIF3, phosphorylated PIF3 makes PIF3 a target for degradation in the proteasome, and once the PIF3 is removed, the repression is removed, and these light response genes are on. And so that is how phytochrome is one example, really, of how phytochrome can elicit one particular response. And if you now go back to what I said at the very beginning, and go back to this summary slide here, a bunch of these things on here we've talked about today, and there will be other things that we haven't talked about. We may get to that, or you may get to those questions as you are thinking about your independent projects and as you are working on your independent projects and later on writing about them. So that's it for today. I know that was a pretty long lecture, but I will see you in lab and I will see you in class. Bye-bye.